Today's session we are going to discuss the Havertz formula. Okay. It is particularly used to draw the ring structures of monosaccharides. When we talk about the representation of monosaccharides, the structure can be drawn. The structures of monosaccharide can be written in two forms. When we draw the structure in an open chain form, also known as Fisher projection formula used to represent that open chain form. And the structures can also be drawn in oxide ring forms. The five-membered oxide ring are known as furanose form, whereas the six-membered oxide ring are referred as pyranose form. So what are the basis of these ring formation? We said that open chain structures of monosaccharide are bent in C shape due to the sp3 hybridization of the carbon atoms. Because of the sp3 hybridization, the carbon have tetrahedral geometry, and therefore, in the open chain structure of monosaccharide, it bends like C alphabet. Okay? And because of that, the certain hydroxy groups at gamma or delta position come closer to the aldehyde or keto group. For example, if we look at the structure of D ribose, it is represented in a Fisher projection here. Okay? And because of the tetrahedral nature of the carbon, we have the C shape. Okay? This is a C shape structure. And what happened? Because of the formation of this C shape, this aldehyde group in the D ribose come closer to this hydroxy group. This is at the gamma carbon, or at carbon number, we can say carbon number 4. So hydroxy at carbon number 4 come closer to this aldehyde. Or in the other, this is a C shape where this aldehyde group comes closer to the hydroxy group present at carbon number 5. So due to this, OH of C4 reacts with aldehyde, or OH of C5 reacts with aldehyde, intramolecular hemiacetal formation takes place, and they gives oxide ring. This oxide ring may be 5-membered or 6-membered. These oxide rings, 5-membered, are referred as furanose ring, as they are similar to the furan form, whereas if they are similar to a 6-membered oxide ring, then it is called as pyranose ring form. The rings similar to furan are known as furanose ring, or called as furanose ring form, whereas pyran are called as pyranose ring form. Now, coming towards the Haber formula, these are named after the scientist William Norman, Sir Norman Harwood. Okay? So that Haber formula demonstrated the cyclic form of glucose acetals, and because of the Haber's contribution in chemistry, okay. So because of the Haber's contribution in the carbohydrate chemistry, he was awarded Nobel Prize of 1937. Now, how these Haber's formula are drawn, or how these ring structures are drawn? We are all familiar with the Fisher projection. Now, let's take an example of D ribose, which is drawn in a Fisher projection. Now, we are going to convert this open chain Fisher projection of D ribose into a six-membered oxide ring form or Haworth formula. That six-membered oxide ring form we also called as pyranose form. So how we can draw a open chain structure to a pyranose form for D-ribose that we will see in a stepwise manner. Okay. So first is we need to draw the carbon skeleton which looks like a six-membered ring structure. Here in this picture you can see I have drawn the carbon skeleton, okay, such a way that it is similar to a six-membered or in a cyclic form. Okay, not exactly cyclic form, but it is a similar kind of structure, okay, which can be then easily drawn or converted into a cyclic form. Now, on this carbon skeleton, now we need to write this substituent. Okay, the carbon skeleton, okay, which is similar to this carbon chain on a vertical line here. Okay, and now we have substituents hydrogen and hydroxy group attached to the carbon number 2, 3, 4. Okay. When we draw these substituent, remember the groups which are present on the right hand side of this vertical line in Fisher projection should be written below the plane. I am repeating, the substituents which are present on the right hand side of this vertical line in the Fisher projection should be written below in the Haworth representation. So here you can notice these hydroxy groups which we have written in a below, okay, or they are below the ring. Next, the hydroxy group present at the C5 carbon react intramolecularly with this aldehyde group and gives hemiacetal or six-membered cyclic form. During this cyclization, a new chiral center is formed. This is the new chiral center formed at C1 carbon, and because of one new chiral center will be having two enantiomers. Now we can say the position of hydroxy group on this newly formed chiral center decide the different isomers. Okay. These two isomers formed, okay, are called as alpha anomer and beta anomer. The isomer in which hydroxy group is below at the chiral center or at the C1 center in the oxide ring are called as alpha anomers. Whereas the hydroxy group which is above at the C1 oxide ring are referred as beta anomer. Due to this intramolecular cyclization of hydroxy at C5 with aldehyde, we get a new chiral center at C1 carbon, which can be written in two isomeric form. The isomer in which hydroxy group is below the ring is referred as alpha anomer, whereas the isomer in which hydroxy group is above the ring is called as beta anomer. Now let's discuss Furanose form of D-ribose. For drawing the Furanose form, start with the Fisher projection of D-ribose and the five-membered ring structure in the Furanose form. So we need to draw the carbon skeleton which looks similar to a five-membered ring form in this manner. Here, during the formation of five-membered ring structure, the hydroxy group which is present at carbon number 4 should be in close proximity with the aldehyde group. Okay. In this representation, the hydroxy group and the CHO group are in the eclipse form. So we can say they are not oriented, okay, in a proper manner to react and form hemiacetate. Now, to have the proper orientation of hydroxy at C4 carbon with respect to aldehyde group CHO, we need to reorient the substituent at C4 carbon. The three groups, namely hydrogen, hydroxy, and CH2OH, will be rotated in a such a way that the stereochemistry at C4 carbon will remain unchanged, and therefore we will get a structure like this. Then the hydroxy reacts intramolecularly with the CHO group and forms the five-membered oxide ring or furanose form of D-ribose. The formation of five-membered oxide ring results in the formation of a new chiral center, and according to the position of hydroxy group attached to that new chiral center, the two isomer are referred as alpha and beta anomer, where the hydroxy group is below and above the plane of the ring respectively. So these two anomers are also called as alpha D ribofuranose and beta D ribofuranose form. Now let's discuss the six-membered oxide ring form of D-glucose, that is pyranose form for D-glucose. This is the Fisher projection for D-glucose. In the first step, the carbon skeleton is drawn in such a way that it looks like a six-membered ring. The hydroxy group present at C5 reacts with aldehyde group in the glucose and form a six-membered oxide ring. But in this particular structure, the hydroxy at C5 position is not properly oriented with respect to CHO. Therefore, we need to reorient the three substituent at C5 carbon, and the structure can be written like this. Here, the stereochemistry at C5 carbon remain unchanged. Then the hydroxy group at C5 reacts intramolecularly with CHO and gives oxide ring formation. These six-membered oxide ring are of two isomeric form, alpha anomer and beta anomer. The alpha anomer is called as alpha D glucopyranose, whereas beta anomer is named as beta D glucopyranose. We are moving next. Now, what are anomers? As we have seen, when the oxide ring forms, we get two isomers or stereomers, okay, and we call them as alpha and beta anomers. So now let's define anomers. The formation of oxide ring creates a new chiral center at the carbonyl atom. The newly formed asymmetric carbon atom is known as anomeric carbon atom. The two diastereomers of monosaccharides, which differ in the configuration only at hemiacetal or acetal, we can say anomeric carbon atom, are called as anomers. The cyclized diastereomers having OH group below the plane of the ring is called alpha anomer, while the one in which OH group is above the plane of ring is called beta anomer. The cyclized diastereomer having OH group below the plane of the ring is called alpha anomer, while the stereoisomer or diastereomer having now we are going to discuss chair conformations. How we can draw the pyranose form of D-glucose in the chair conformation. We all know that the six-membered cyclic ring having a more stable conformation, that is the chair form or chair conformation. The two anomeric form of D-glucopyranose are written here, which then easily converted into the respective chair conformations. Here we have two chair conformations for alpha D glucopyranose and beta D glucopyranose. The subsequent, which are written above or below in the Haworth formula, will be written in the same manner in the chair conformation. That is, the OH, which is below in the alpha anomer, is written below in the chair conformation. Similarly, for the other hydroxy and CH2OH group. For the beta anomer, the hydroxy group, which is above the plane of this cyclized oxide ring, then the six-membered chair conformation, it is written above.